Hello, everyone, and welcome to today's live broadcast, Guiding Pancreatic Cancer Therapy by Individualized Epigenetics. I'm Alexis Corrales of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Agilent. For more information about our sponsor, please visit their website at agilent.com. Now, let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the button. If you have trouble seeing or hearing the presentation, please click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I will now turn it over to Dr. Valentina Maran to introduce our speaker and our presentation for today.
Thank you for the introduction. I am Valentina Maran, Global Product Manager for Microarrays at Agilent Technologies. Today, I'm happy to present the great work performed by Dr. Surajit Dhara, Senior Research Scientist at the Dartmouth-Hitchcock Norris Cotton Cancer Center. Dr. Dhara's team have developed an individualized epigenetic tool to help guide pancreatic cancer therapy through the analysis of individualized epigenetics. This is a great example of what can be achieved thanks to the customization capabilities of our microarray platform. Now I leave the microphone to Dr. Dhara.
Thanks, Valentina, for your kind words. And I am Surajit Dhara. I am the co-founder and president of Episteme Prognostics, which is a precision medicine company, a spin-off from MSKCC in New York and Dartmouth-Norris Cotton Cancer Center in New Hampshire. So what we do, we try to guide pancreatic cancer therapy by individualized epigenetics. And I'll take you through our work, which we recently accomplished. First slide. So we focused pancreatic ductal adenocarcinoma, which is the most lethal form of pancreatic cancer, and pancreatic cancer is one of the most lethal malignancies known till date. And this is projected to be the second largest cause of cancer-related death in the United States by 2030. There are over 57,000 new cases in the United States and 500,000 worldwide recorded in 2020, and these numbers are rising fast. So what happens in clinic? So this is the standard protocol in a PDAC clinic. Any patient comes with a suspected malignancy, they're radiodiagnosed, followed by biopsy confirmation of the lesion, and then stratified to 3 groups: resectable, borderline resectable, and non-resectable for therapeutic intervention. The resectable group, they qualify for upfront surgery followed by an adjuvant chemotherapy. The borderline resectable group, they first undergo a neoadjuvant chemotherapy to downstage the tumor followed by surgery. And the non-resectable group, they only get chemotherapy. No surgery is involved in this group. So what we can see in the best case scenario is about 20%, 20 to 30% response. When I say response, meaning long-term response, long-term survival, disease-free for more than 5 years. As shown in this cartoon, that resected tumors are resected patients, that primary tumor is completely resected by a process called Whipple, as shown in this cartoon on the left side of the slide. And the Kaplan-Meier graph that shows what we see in the clinic after follow-up. So as you can see here in this graph, that post-resection, all patients are apparently disease-free, and they were put on 2 adjuvant chemotherapies. Green, the blue line is gemcitabine, and the red line shows FOLFIRINOX, 2 different chemotherapy. And as time progresses, in x-axis you can follow, within a year, you see a significantly large number of patients, close to half, they relapse. Some more patients relapse within 42 months or around 42 months, but this 20 to 30% patient at the right side end of this Kaplan-Meier, they live for more than 5 years disease-free. So the take-home is virtually all patients are treated with either FOLFIRINOX or gemcitabine in pancreatic cancer, and which does not include any molecular markers predicting response. And the result that we can see is 70 to 80% patient relapse, unfortunately. So we hypothesized, we wanted to understand the nuances. So we hypothesized epigenetic differences at the level of chromatin accessibility, which potentially linked to distinct differentiation states, might distinguish rapidly recurring from non-recurring tumor. And I'm going to explain that soon. So before that, first rationalize why epigenetics. Because number one, epigenetics correlates with phenotypes which genetic mutations do not explain. For instance, 37 trillion cells of our body, they share identical genome, identical DNA sequence, but it has been proved that they're epigenetically different. All biological aspects have been correlated well with epigenetic patterns. Number two, it is robust. Unlike gene expression signature, you can avoid the RNA-related tissue RNA degradation issues. And third is epigenetic reprogramming has an enormous potential. I can talk about it day long, but it has its powerful impression in the health sciences field, from regenerative medicine to cancer. So considering these 3 facts, we thought to understand the nuances of outcome of PDAC in the light of epigenetics. The second rationale is why chromatin accessibility. Chromatin accessibility summarizes all known upstream epigenetic mechanisms. For instance, 10 years before, we knew epigenetics means DNA methylation. But now we know epigenetics is more than that. Epigenetics encompasses DNA methylation, histone modification, and chromatin remodeling. So finally, all the mechanisms upstream leads to a right balance, maintaining a right balance of open and closed chromatin. So now, with this background information, I'm taking you through our experimental details, how we accomplished our goal. So we tested our hypothesis by investigating tumor-intrinsic chromatin accessibility landscape of PDAC. Because the first challenge that we encounter is tumor microenvironment is notoriously heterogeneous. Like all other tumor, PDAC is also notoriously heterogeneous. It has epithelial, malignant epithelial cells, fibroblast cells, endothelial cells, and other cells, and infiltrated immune cells. One more thing, as depicted in figure B, which is unique to PDAC. If you take a resected piece of tissue from PDAC tumor, the epithelial cellularity notoriously varies. you may not get a single cell in that, versus 90% of the tissue will be full of epithelial cells. So we wanted to see the tumor-cell-intrinsic epigenetic pattern in these PDAC tumors. So what we did, we tried to physically isolate the malignant epithelial cells by epithelial cell adhesion molecule conjugated magnetic beads, which is simpler to isolate. And as it's shown in this cartoon, that we have isolated epithelial EpCAM positive and EpCAM negative cells from the resected tumors. And then we compared these two subpopulations to make sure that we have enriched tumor population by KRAS mutant allele frequency enrichment and microarray analysis, gene expression analysis between these two subpopulations, as shown in the lower panel C and D. And after we are sure that our enrichment is satisfactory, we went ahead and did ATAC sequencing on EpCAM positive subpopulation of PDAC tumors. We sampled replicate samples from every tumor. How? Any piece of tumor that we get, we allocate it into two, and then independently process them. Transposition reactions and all other reactions are independently done. So although they were not biologic replica in every tumor, but they are not technical replica either. So they are like in between biologic replica and technical replica. Just to make sure, just to be very sure about what we discover is correct. So what we did is every patient, each and every patient, two replica as shown in figure A are plotted like in X and Y axis, and we took the overlapping peaks. And this first panel on the left side shows like a lot of satisfactory overlapping peaks in the dark black region, while the left side panel shows a very little, tiny little black region. That means this particular patient we didn't retrieve many reproducible peaks, maybe because of our technical artifact. So what we did, we took the best quality libraries selected by, this is called IDR, irreproducible discovery rate, selected by IDR. So we took the best quality 40 patients library for our discovery set analysis. And we tested if 40 patients are good enough to cover the entire repertoire of open chromatin peaks in human pancreatic ductal adenocarcinoma. So as you can see, it nearly reached to saturation. That means we have discovered this 121,697 open chromatin peaks from 40 PDAC patients. And if we increase, say up to 400, extrapolate, we may have included some more peaks, but that may not be worth. However, with this entire human PDAC reference epigenome, we identified a specific signature of 1,092 regions which were represented differentially between recurrent group of patients and non-recurrent group of patients as shown in the figure C. So in this figure, you see the red regions on the top are 300 regions which are open in patients who are recurrent, and 700 regions in the bottom, the green slab region, which are silenced in these patients. So for instance, this is one of the example, TASC3 promoter, tumor suppressor candidate gene 3 promoter region as depicted for an instance. That this particular promoter is silenced in recurrent patients. The red tracks on top shows the genomic track retrieved from the recurrent patients, and the blue track shows the same from non-recurrent patients. And the lower panel here is ENCODE-defined TASC3 promoter region. As you can see in HPDE6 at the bottom, that TASC3 promoter is open, which is a normal pancreatic cell line. Whereas in aggressive pancreatic cancer, the task3 region, the same region here, is silenced, closed, which is further corroborated by DNA methylation profile. This is ENCODE data. Now we overlap this ENCODE data with our data in the same region. As you can appreciate that almost all red tracks, this particular region, task3 promoter region, is silenced. Whereas the blue region here, almost all patients, this region is opened, not silenced. And we have 1,092 such regions in the genome that we discovered are represented differentially between patients who recur versus patients who did not recur. Then we further thought, so these open and closed regions, whether they represent what transcription factor or regulatory elements, what transcription factors they bind to their regulatory elements. And we discovered through a motif search that 61 transcription factors which represent differentially between patient who recurred, which is shown in the top red panel in figure A, and the motifs which represent in non-recurrent patients, which is represented in blue. And then we further confirmed with immunohistochemistry the top hits, HNF1 and ZK scan as shown in figure B. As you can see here in figure B, in a representative recurrent patient, HNF nuclear localization of HNF is completely abolished. Whereas in a non-recurrent patient, very starkly contrasting green dots you can see in the nuclei. And the ZK scan showed an opposite trend. And with HNF alone, you can segregate patients based on their disease-free survival. Then we went ahead and validated these results to another completely independent cohort of 97 PDAC tumor tissue microarrays, where 45 patients were non-survivor. They lived, their overall survival was less than 6 months, whereas 52 patients were long survivors. They lived over 6 years as median overall survival. And you can appreciate from this figure, in figure D, that in all short-term survivors, the nuclear localization of HNF is completely gone, completely absent. But you can see sharp dots, red dots in the nuclei of long-term survivors. And you can see them in the quantitatively in figure E. So after this validation, we went ahead and thought that, well, our invention, our discovery of this 1092 signature, we extrapolated to clinical diagnosis. But ATAC-seq being a cumbersome, expensive method, and we really reasoned that this chance of its translation into every clinic will be difficult. So we invented ATAC-array, the only microarray that reads chromatin accessibility. So now this is a busy slide. I'll take you through our invention. What we did is a very simple way, but very effective and robust method of reading chromatin accessibility. So what we reasoned that if we print the differential regions on a glass slide, on a microarray, as shown in this cartoon, the left side red group of humanoids are recurrent group of patients, and the blue are non-recurrent group of patients. And they have signature, which is depicted here simply, that red block here are 300 regions which are differentially open in recurrent patients, whereas the blue regions are silenced and vice versa, opposite. And the green regions are open in both the groups as positive controls. So if we have an ATAC library made from this group of patients, the red group of patients or recurrent group of patients, the red region in the library will be predominantly amplified, whereas the blue regions, since they are silenced in these patients, they will not be represented. And the opposite profile you could expect from an ATAC library of a blue group of patients, or non-recurrent group of patients. So now, if we hybridize these libraries with the microarray, we get these differential profiles as shown in the bottom two figures. In the left side, see the red peak here is shifted to the right of the blue peak, as we expect it would be, because the red region here is predominant. So intensity coming from red regions are more than the blue regions. And green as a positive control, the most intense regions, and the black peak here is a negative control provided by adjuvant as CG backbone of the microarray. So that's what you would expect. And the exactly opposite profile you can see from a patient who were non-recurrent. So these two are the real data from two real patients. And we found that within these 932 regions, which was covered by adjuvant probes out of 1,092 regions, they remarkably correlated with ATAC-seq confined to these regions. And that was really remarkable correlation. So in other words, ATAC-array does the job of ATAC-seq, but in a lot convenient way. And you can print any signature and conveniently read on ATAC-array. And also, we IP'd our candidate motifs, including HNF. So now we developed a combined tool called Episteme DX, which is actually a combination of the prognosis score derived from our ATAC array and the nuclear localization patterns of HNF. So together, we could decipher the nuances of PDAC, and we see that it did segregate patients into significantly different DFS groups, as shown in figure B, which is ATAC array alone could segregate them into two groups. And when you combine ATAC array with the HNF, so calling it Episteme DX, our segregation is way more confident. And we could segregate these patients into 4 distinct groups. And we also validated in an organoid cohort, which is shown in figure D. So Episteme DX works beautifully. So now the value of this Episteme DX is if we can include them in the current clinical diagnostic procedure. So if patient comes as they normally get radio diagnosed and biopsy diagnosed at the clinic for suspected malignancy, if we include Episteme DX in the whole process, and we get result in 3 days. So that means we can utilize the time window of our standard diagnostic process. And at the end, the oncologist will know if a patient carries a good prognosis or a bad prognosis signature. If a patient is upfront resectable but carrying a good prognosis signature, they can straightway go for upfront surgery and adjuvant chemotherapy. Their chance of long-term response becomes 100%. Whereas a patient, although qualifies for surgery, but having a bad prognosis, now we know these patients are the ones who are going to relapse if we follow this adjuvant path. So we can think of an alternate therapy for them, either by neoadjuvant path right at this moment, this is currently a big deal to take a decision between adjuvant versus neoadjuvant path for PDAC patients. And also, we can combine an epigenetic drugs. And in few slides, I'll explain why epigenetic drugs, because our profile is epigenetic profile. So the value proposition that we have is oncologists can better stratify the therapy, and also with epigenetic drugs, we can develop companion diagnostics with PanCAPisteme DX. So now I suggest why we can propose an epigenetic therapy and tailored epigenetic therapy to PDAC patients who have poor prognosis signature. And this is a hypothetical, it has to be tested, but this is a pretty testable hypothesis. So this is a snapshot of our signature. And two of them I already explained, TASC3 and HNF. And we now recently known that HNF silencing is done by enhancer of Gesta homolog 2 is a component of polycomb repressor complex. So now we know EZH2 silenced HNF. So there are many drugs available at this moment which we could utilize to reprogram the epigenome. So this is a hypothetical scenario. So if we have a patient with a bad signature, as shown in the upper panel of this cartoon, the patient refractory to chemotherapeutic agent will have a profile like that. The red peak will be shifted to the right of the blue. So if we give an epigenetic drugs such as ATAC inhibitor or tazemetostat, like EZH2 inhibitor or so on, what we could expect is opening up of the silenced regions, and the intensity will move to right on top of the red peak. And we can expect a better response because we have reprogrammed the epigenome. Whereas a patient who already have a good prognosis signature, if we know beforehand, we do not, first of all, you do not need to give them any epigenetic drug. They are already good. They are 100% sure they'll respond. They'll live for more than 5 years disease-free. So we may not give them. If we give, we may not increase their chances of response to chemotherapy. However, this is a testable hypothesis, and we are testing it through a proper pathway as shown in the lower panel till clinical trial. So we have right now 9 epigenetic drugs and many more in the pharma pipeline for us to develop companion diagnostics with PanCAPisteme DX. So to summarize, is the last slide, I believe. To summarize, our Episteme development pipeline is, number one, we are developing it from an invention of an academic lab to a cancer diagnostic kit in hospitals, so that all hospitals and path laboratories can use it for their routine clinical stratification. We are optimizing all different aspects of benchmark measures of the ATAC-array and testing its feasibility in liquid biopsies. And also, number two, as I explained, we are also planning to develop companion diagnostics with epigenetic drugs through a formal path from cell line to clinical trials. Although our main focus has been pancreatic cancer, but of course, potentially it is applicable to other indications as well, but it's a long way to go again. And with this, I conclude my today's presentation and acknowledge the team. First of all, I apologize for the names which are not included here, because there are many people who are directly or indirectly involved to accomplish this project, to bring this project to success. But the names here, which were directly interacted with, and the author in the author list of the manuscript. So it involves multiple institutions and adjuvant technologies, of course. Without them, it would have not been possible. And also, I thank Agilent for giving me this podium. And I thank you, Alexis, for conducting all this from LabRoot's side, and thank you, LabRoot, for doing this. There's a disclosure to give. Right now, Agilent products are not for diagnostic procedures; they're research use only. And that's it. And that's all I have for today's presentation. If you have any question, I'll be happy to take. I realize that although I mentioned in the beginning that Episteme Prognostics is the spin-off from MSKCC and Dartmouth, but this is the formal disclosure which says most of the research work was done in MSKCC, Dartmouth, and the other collaborative institutes under the leadership of Professor Stephen D. Leach. He was the former inaugural director of David M. Rubenstein Pancreatic Cancer Center, Memorial Sloan Kettering Cancer Center, and currently the director of the Dartmouth-Hitchcock Norris Cancer Center. And Steve is also the co-founder of Episteme Prognostics. And also, I would like to especially thank Valentina, Maran, and Carol Koch for helping me while preparing for this webinar. Thank you, Carol. Thank you, Valentina. And I can ready to take all the questions that you have.
Thank you. Thank you so much, Dr. Dhara, for your informative presentation. So we will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Dr. Dhara, our first question. How is the ATAC-array advantageous over ATAC-seq?
All right. So first of all, these are two different technologies. We are not competing with ATAC-seq. ATAC-seq is more of a discovery tool when you want to know the de novo new discovery of a signature. But once you discover a signature, like our 1092 signature, and you want to employ it in a diagnostic purposes, then ATAC-array is simpler, faster, cheaper than doing ATAC-seq. So that's how advantage of microarray could be leveraged.
Wonderful. Thank you so much for that clarification. So Dr. Dhara, moving on to our next question, a little similar to our first one, which is how are epigenetic signatures advantageous over gene expression signatures?
Well, so this is a good question, first of all. So we know that now through all these international consortiums in last decade, most of the malignant diseases have been subclassified into molecular subtypes, and which means that the gene expression signatures differ from each other with different prognostic groups. But the problem is these signatures, gene expression signatures, often comprise of multiple genes, which that means if you have a specific pattern of a gene expression in a typical poor prognosis patient, technically you should be completely reprogram them, and it is virtually impossible to target all the genes. Whereas if you see an epigenetic signature, the collective reprogramming of the regulatory element by an epigenetic drug is possible. So for in our case, just explained, 700 regions were silenced in an epigenome of a poor prognosis patient. It could be opened up by one or two drugs like HDAC inhibitor or tazemetostat that I explained. So that's how the collective reprogramming opportunity it gives. That is why it is more advantageous than gene expression signatures.
Wonderful. So, Dr. Dhara, is the 1092 chromatin signature causal to chemotherapy response in PDAC?
Well, that part we are testing at this moment. as I showed through hypothetical cartoon slides, that if they were causal, then we could, if we open them, then they would be making the tumors amenable to chemotherapy. And if they do, then yes, they're causal.
Wonderful. So just as a reminder to our audience, those questions that we are unable to answer today and those that come in during the on-demand period will be answered by the speaker via the email address that you provided at the time of registration. So please continue to input your questions for Dr. Dhara today. So moving on to our next one. Dr. Dhara, will it be possible to apply the same array and or approach to a different cancer type?
Yes, of course. So two ways you could be doing it. Number one is testing how many of among these 1092 overlap with other tumor types. Or you can have a complete discovery of cancer-specific signatures by ATAC-seq, the way we did in PDAC, and then print that signature to ATAC-array for diagnostic purposes. So yes, it is extrapolated. It can be extrapolated.
Thank you so much. And looks like we've got one more question for you. So, Dr. Dhara, how can we access this technology?
So one of the business strategy that Episteme right now we are thinking is a paper service. Is one of the, in addition to developing it as a kit, in addition to developing Episteme DX as a companion diagnostics, we also do paper service. If you're interested to get the profiles done by ATAC-array, we can do it for you. We are commercializing this technology.
Perfect. And it looks like we've got another question that popped into here for us. So how long, here it is. How long do you think it will take for these technologies to be included in diagnostic procedures?
So this is actually, it has to be done through clinical studies, correlative clinical studies. So follow-up of at least 3 years. So overall, it will take 3 to 4 years to actually bring it to the FDA-approved, 510K approved diagnostic kit.
Okay, perfect. Thank you so much for answering that. So we still have time for a couple more questions. So if any audience members want to submit their questions, it's in the far left-hand side of your studio. More than welcome to submit questions to Dr. Dhara. But do you, while we're waiting for some questions to come in, Dr. Dhara, do you have any final comments for our audience?
Not really. And all that I wanted to tell you is thank you all for attending this webinar. And please feel free to write me an email as displayed in my email address, surajit.dhara@epistemeprognostics.com. I'll be happy to answer your questions.
Perfect. Thank you so much, Dr. Dhara. So looks like that is all the time we have for today, but before we go, I would like to thank the audience for joining us today and for their interesting questions. As a reminder, questions that we did not have time for today and those submitted during the on-demand period will be addressed by Dr. Dhara via the contact information that you provided at the time of registration. We would like to once again thank Dr. Dhara and also Dr. Maran for their time today and their important research. We'd also like to thank Lebanon and our sponsor, Adjuvant, for underwriting today's educational webcast. You can view the webinar on demand. Lebanon will alert you via email when it's available for replay. That's all for now. Thank you so much, everyone, for joining us, and until next time. Bye-bye.
Thank you, Alex. Bye-bye.